We're going to start. Today we're going to continue discussing cells, and we're going to talk about eukaryotic cells for a while. Now, keep in mind, eukaryotic cells are found in plants and animals and fungi, which include things like mushrooms, molds, yeast, and there's another group of eukaryotic organisms that have eukaryotic cells, and these are something called protists. And protists aren't really a very good group. They're kind of the leftovers. They're things that have cells that have a nucleus, and that's what eukaryotic cells have. But they don't really fit with animals or plants or fungi. So we sometimes put them in a separate group of their own, protists. Most books actually divide protists up into 10 or 20 or 30 different categories because it turns out that there are really a lot of small groups of protists that are very different from each other. So let's focus to start with on the nucleus of a eukaryotic cell. And again, we're going to start out thinking about how a eukaryotic cell makes proteins. So just like with prokaryotic cells, really the story of proteins in a eukaryotic cell begins in the nucleus. So the nucleus is our large circle in here. Keep in mind that most of the cell is out around the nucleus, and it's not shown in this diagram. So these little kind of blue channels that you can see coming out from the nucleus are something called the endoplasmic reticulum. Long name, but endoplasmic means that it's in endo, the cytoplasm, plasmic. And reticulum is just a network. There's a group of blood vessels underneath your brain that are called a reticulum. Because they form a kind of a circle underneath the brain. But let's focus on what's inside this kind of inner blue area. So this outer line around the purple, and then there's another line outside that, kind of around this first blue. This is a membrane. Now, each line is a membrane. So there are actually two membranes around the nucleus. We'll see some other organelles later that are separated from the cytoplasm by a double membrane, but the nucleus is one. So there's an inner layer to this membrane. There's an outer layer, and the outer layer is actually a part of this endoplasmic reticulum. Now, this endoplasmic reticulum has some little red dots on it, and the little red dots represent ribosomes. So if you would look at this under a very good microscope, you'd have to have a microscope much better than the ones that we have at school. But if you would look at this under a microscope that allowed you to see the ribosomes, because of the ribosomes on the outer surface of the endoplasmic reticulum, it would look kind of bumpy. So it's sometimes called rough endoplasmic reticulum. So we'll talk about that more later. So we have a nuclear envelope. Every once in a while in the nuclear envelope, if you look, you can see little indentations or little holes that go through the nuclear envelope. These are nuclear pores. This is how things get in and out of the nucleus. Now, on the inner side of the pore, there are some proteins. And the proteins kind of work like a bouncer at a club. They decide who gets into the nucleus and who gets out, except the way they do it doesn't depend on, how old you are or anything like that. Depends on the chemistry of the molecules. And molecules coming into the nucleus have a certain type of chemical shipping tag put on them. and molecules going out of the nucleus have a different type of shipping tag put on them. So things are going to move in and out of the nucleus through these pores. Now, what's actually in the nucleus? Well, three main things that we're interested in. There is some material inside the nucleus that's kind of part solid, part liquid, similar to what we talked about with the cytoplasm yesterday. It's kind of like a milkshake type consistency. Since it's in the nucleus, it's called the nucleoplasm. Now notice, the membrane around the nucleus, the double membrane, is called the nuclear envelope. The fluid inside is the nucleoplasm. The holes in the membrane are the nucleopores. I think you're starting to get the idea now. If you ever saw that old Batman TV show where everything was named the Bat something, you know, they got to the Bat Cave by sliding down the Bat pole. Everything connected to the nucleus is the nucleus something or other. So kind of get used to that idea. Inside the nucleus, there's a smaller dark circle. Some cells inside their nucleus have more than one of these small dark circles. This is called the nucleolus. That OL kind of towards the middle of the word means little. So when people first started looking at the nucleus through a microscope and they could see the nucleolus, it kind of looked like a little nucleus, because the nucleus was a dark circular area in the middle of the cell. The nucleolus was an even smaller, darker area in the middle of the nucleus. Now the nucleus is where the cell is going to start building RNA, especially the RNA that's used to build ribosomes. You might remember the other day we said ribosomes are built from protein combined with rRNA. The rRNA, the first R stands for ribosomal. So this is where the cell starts building ribosomes. It doesn't build them completely, it just barely gets started, because complete ribosomes, these little red dots, you can't tell from the scale of this drawing, but they're actually too big to get out through the nuclear pores. So they're put together partially, they're shipped out of the pores, and then they're assembled later. So inside the nucleus, we've got this nucleoplasm. Somewhere we've got this nucleolus, maybe more than one. And the other thing, and in a sense, the crucial part, is we have chromosomes. Now remember, the bacteria cells, the prokaryotic cells that we looked at the other day, only had one chromosome. And if you remember, we said there were three crucial things that we wanted to remember about this chromosome. One was the fact that there was just one chromosome. Another was the fact that it was a circular molecule. Chromosomes have DNA in them. The DNA is made of nucleotides. In a circular molecule, the last nucleotide in the DNA is chemically bonded back to the first nucleotide. So it's like a bunch of people holding hands playing Ring Around the Rosie. So the molecule's circular, there's only one of them, and we said it's naked protein. There's naked DNA, I'm sorry. There's no protein associated with the chromosome. Now, if we look at the chromosome in a eukaryotic cell, and for instance, you have eukaryotic cells, what we see is they're different in every way. Where bacterias only have one chromosome, and that means they reproduce asexually without a partner. Eukaryotic cells almost always have an even number of chromosomes. People have 46, chimpanzees have 48. There's one type of lily that has 96. There's one type of rabbit that has 44. There's one type of snake that has 18. Why do eukaryotic cells almost always have an even number of chromosomes? Because they can reproduce sexually. And when they reproduce sexually, they get one set of chromosomes from mom, one set of chromosomes from dad. 2 times anything is an even number. So that's why eukaryotic cells tend to have an even number. There's a few weird exceptions that we'll talk about later, but they are kind of exceptions to the rule. Now, not only do people have an even number of chromosomes, 46, or you could think of it as 23 pairs, since you get one of each pair from your mother, one of each pair from your father. Not only do people have 46 chromosomes, but our chromosomes are linear. The first nucleotide is chemically bonded to the second nucleotide, the second to the third to the fourth. And people have an average of about 150 million nucleotides in a molecule of DNA. When we get to the last nucleotide, it's not bonded back to the first. That's just the end of the molecule. So it's very similar to when we talked about carbon. We said carbon could make straight chain molecules. That's similar to what we see in a eukaryotic cell, or it could make rings or cyclic compounds. That's similar to what we see in a prokaryotic cell. Now, there's a third difference. When we look at the DNA in your chromosomes, compared to a bacteria cell, your cells are bigger, but they're still microscopic. They're still pretty small. But they've got a lot of DNA in them. Even though a typical cell is microscopic, if we would take all the chromosomes out of your cell and we would lay them end to end, they would stretch about 3 feet. So you've got 36 inches or so of chromosomes in a eukaryotic cell that's microscopic. So that's a lot of chromosomes in here. So somehow we have to organize these chromosomes so they don't get tangled up. And this is where the third difference between eukaryotic chromosomes and prokaryotic chromosomes comes into the picture. If we look at eukaryotic chromosomes, they are made of DNA, but the DNA is wrapped around proteins, kind of like thread is wrapped around a spool. And what this does is this helps to organize all this DNA. Since there's so much DNA in such a small space, this helps to prevent it from getting tangled up. Doesn't work perfectly well, you know, what does? But it does a pretty good job of keeping the DNA from getting tangled up. Now, just like we saw in prokaryotic cells, if we look at the DNA in the chromosomes, the DNA can be divided up into short segments, and these segments are called genes. Each gene is the instructions for the primary structure of a protein. In other words, the order of the amino acids in that protein. Now, you have a few more genes than bacteria cells do. Bacteria cells have about 10,000 genes. So that means a bacteria cell can typically make about 10,000 proteins. If we would look at one of your cells have about 22,000 or 23,000 genes. We're not sure about the exact number right now. But even though you have twice as many genes as a bacteria cell, if we take one cell out of your body, doesn't matter where that cell comes from, your brain, your skin, a red blood cell, a bone cell, a liver cell, wherever we take that cell from, it makes about 10,000 proteins. So even though there's 22,000 genes in the cell, it only uses about 10,000 of those genes to make proteins. Now, the difference between you and a bacteria cell, if we look at a bacteria like E. coli, and we look at a big cluster of E. coli, thousands of cells, they're all using the same 10,000 genes. If I take a liver cell from you and a skin cell and a heart cell and a brain cell, they're not all using the same genes. They have 23,000 genes to pick from, if you want to think of it that way. Now, which genes they pick and use to make proteins? Well, that's the difference between a heart cell and a brain cell and a kidney cell, muscle cell. So we're going to use a gene in here, and we're going to make some mRNA. Remember when we talked about mRNA, we said M was messenger RNA. And the messenger RNA was a copy of the instructions on the DNA. Now in a bacteria cell, we didn't have this nuclear envelope. So as soon as we made messenger RNA, the ribosomes came in right next to the chromosome, attached to the messenger RNA, even before it was completely made, and started making a protein. But here, since the ribosomes are out here, the ribosomes are outside of the nuclear envelope. The chromosomes are inside the nuclear envelope. When we use the chromosomes, when we make some messenger RNA, we have to send the messenger RNA out of the nucleus to the ribosomes, and then we can make a protein. Now, there's a disadvantage to this. This is a little bit slower. A bacteria cell can make proteins much more rapidly than a eukaryotic cell can. Prokaryotic cells can make proteins almost instantly. But there's also an advantage. This method of making proteins is a little bit slower. Why? Because we have to ship the messenger RNA all the way from the chromosome out to one of these ribosomes. But the advantage is this means that eukaryotic cells can just hold on to their messenger RNA in the nucleus. They can wait a while. They can see if they want to be certain that they want to make that protein or not. So this gives eukaryotic cells a finer degree of control over whether they make proteins. For bacteria cells, for prokaryotic cells, as soon as they make messenger RNA, they make the protein about 99% of the time. If we look at human cells, when your cells make messenger RNA, they only send it out to a ribosome and make a protein from it about 20% of the time. About 80% of the time when your cells make messenger RNA from the DNA, it stays in the nucleus and nothing happens to it. After a while, the cell decides, we don't need that protein anyway. So it just takes apart the messenger RNA and recycles the nucleotides. So just like in bacteria cells, the chromosome, or if you want to think of it in bigger terms, the nucleus, the gene on the chromosome, this is the instructions to make our protein. So this is where the whole process of protein synthesis starts. And remember what we've said, cells are factories that make proteins. So this is protein synthesis is the major activity that cells do. So let's think about ribosomes now. There are thousands of ribosomes in a eukaryotic cell. In this drawing down here, the ribosomes are the little dots in here. This is rough endoplasmic reticulum. So some ribosomes are attached to the endoplasmic reticulum, but if you look, you can see some of those same little dots over here or up in this kind of upper right-hand corner of the cell. Some ribosomes aren't attached to endoplasmic reticulum. So ribosomes can be attached to endoplasmic reticulum. That's what we kind of see on this beige tan area here. Or ribosomes can be attached to something we'll talk about a little bit later called the cytoskeleton. Cyto means cell, skeleton, just like you, that's the framework of the cell, just like the skeleton is the framework of your body. So ribosomes can either be kind of attached to the cytoskeleton, or ribosomes can be attached to the endoplasmic reticulum. Now, it doesn't really matter which they're attached to. Either way, they make proteins. Now, remember what we said, ribosomes are made up of two parts. They're made up of a large subunit and a small subunit. The large subunit here is 60S. The small subunit is 40S. If you look back at our discussion of prokaryotic cells the other day, the large subunit was only 50S and the small subunit was only 30S. So notice these are bigger than the parts of a ribosome in a prokaryotic cell. And the entire ribosome is also bigger. So there are some antibiotics like tetracycline that affect the ribosomes in prokaryotic cells, but not in eukaryotic cells. Because of the fact, remember, as we keep saying, everything on a molecular level works by its three-dimensional shape. So the ribosomes in a prokaryotic cell are different sizes, and that means different shapes than in a eukaryotic cell. So a tetracycline that interferes with the ribosome in a prokaryotic cell, leave your ribosomes alone. So there's an antibiotic called tetracycline, and it interferes with the ribosomes in prokaryotic cells, making proteins. It doesn't interfere with human ribosomes. Now, there are some people who are sensitive to tetracycline, but they're not sensitive to it because it interferes with their ribosomes. They're sensitive to it because it can interfere with a different molecule. So both of these subunits have the rRNA. That's the RNA part of the ribosome, and they both have protein. So the second step in making a protein in a eukaryotic cell, the instructions are on the genes, which are part of the chromosomes in the nucleus. And then we make a copy of those instructions, messenger RNA, and we send that out to a ribosome, and the ribosome makes the protein. Now, keep in mind what we said the other day. A prokaryotic cell is only about as big as this little oval structure here that's blue or purple. And this is called a mitochondrion. So when we make a protein in a prokaryotic cell, it never has very far to go. But in a eukaryotic cell, we could make a protein in a ribosome up here, and we might have to use it way out on the plasma membrane. So this means that we've already made our proteins, but now the rest of protein synthesis is concerned with getting that protein where we're going to use it. So this is something we didn't really have to worry about in a prokaryotic cell, but in a eukaryotic cell we do. So this is where the endoplasmic reticulum comes in. So here's kind of a three-dimensional view of the endoplasmic reticulum. Notice on the inside, we see our nucleus, and we see that nuclear envelope. And notice that the outer layer of the nuclear envelope, remember what we said, is continuous with the endoplasmic reticulum. So we have two types of endoplasmic reticulum. The endoplasmic reticulum closest to the nucleus has ribosomes attached. And as I said, if you look at it under a microscope, it looks like it's got little bumps on it. So this is called a rough endoplasmic reticulum. The endoplasmic reticulum that's further out, that's closer to the plasma membrane, to the outer edge of the cell, doesn't have ribosomes attached. Because it doesn't have ribosomes attached, if you look at it under a microscope, the edges look nice and even. So this is called smooth endoplasmic reticulum. Now, the smooth endoplasmic reticulum on its outer edge, it does have some enzymes. And these are enzymes used in making lipids, especially the phospholipids that are found in the membranes of the cell. Now, notice that all these black lines that we see here, these all represent membranes, or if you think of the kind of the blue or the blue grading into orange and then some pink, these are all membranes. If we look at the ribosomes, they're attached to the outer part of these membranes, but sometimes these membranes of the endoplasmic reticulum look more like pipes. They're kind of rounded. Sometimes they look more like kind of a flattened plastic sac. But either way, these endoplasmic reticulum in the cell, they don't just attach the ribosomes to make proteins. They don't just attach enzymes to make lipids. If you go inside the endoplasmic reticulum. This is a transportation system. It's kind of like if you've ever seen that cartoon show Futurama, where at the beginning of the show that shows the people zipping through those little tubes. Proteins and other molecules can travel through the endoplasmic reticulum to move from one part of the cell to the next part of the cell. So this is a transportation system. Now you might wonder how molecules, how does the cell know where a molecule goes? How does it know if it goes to this kind of pinkish red area or part of this blue area? Well, there are chemical shipping tags put on all the molecules. And there are proteins that can read those chemical shipping tags. And once they read them, they work kind of like forklifts. And they carry the protein to the part of the endoplasmic reticulum where it's supposed to go. Now, one of the main places that proteins get transported to is something called the Golgi apparatus. And the Golgi apparatus is another membrane-bound organelle. So notice, so far today, we've talked about the nucleus. That's a membrane-bound organelle separated from the rest of the cell by its own membrane. We've talked about the smooth and rough endoplasmic reticulum. Those are membrane-bound organelles. They're surrounded by a membrane. The Golgi apparatus is also a membrane-bound organelle. This has been kind of sliced in half so that we can see inside it. And it's kind of similar to the endoplasmic reticulum. It reminds me of kind of a stack of paper bags that you might have sitting someplace before you take them to recycling. So there's a little hollow area inside. So a protein is going to get carried to the Golgi apparatus. When it gets close to the Golgi, the endoplasmic reticulum and the Golgi don't actually touch. But when the protein gets as close to the Golgi apparatus as it can get, it pinches off in a little blob of fat. Kind of like if you've ever seen in a frying pan where you have a little drop of oil pinch off from the rest of the oil. Here, this drop of oil, the outer edge, is phospholipids. So this is another membrane-bound organelle. Inside is the protein that started out in the endoplasmic reticulum. And this is called a transport vesicle. And we're going to carry the protein from the edge of the endoplasmic reticulum to the Golgi apparatus. And the transport vesicle always drops off the protein on the cis face. And the cis face is just the edge of the Golgi apparatus that's nearest to the nucleus. So the protein gets dropped off, and when it gets dropped off, now it's in this first layer, in this kind of darker purple in the inside. And in every layer of the Golgi apparatus, there are enzymes. And what these enzymes do is these enzymes change the protein chemically. You could think of it like an assembly line, where when they're building a car, one spot puts on the tires, one spot adjusts the steering wheel, another spot puts the doors on. Here, some of the enzymes add things to the molecule, some of the enzymes take things away. Now, some proteins get dropped off at the second or third layer, and that's because each layer has different enzymes. So depending how a protein has to be modified, it doesn't always go through all of these layers. It might start at the second layer on the cis side instead of the first, but it gradually moves in this direction towards the trans face. And the trans face is the side closest to the plasma membrane, closest to the outside of the cell. And whenever we get done with all the modifications, it might be in this last layer, but it might be before that, a little droplet of fat pinches off again. So this is another phospholipid surrounded vesicle. The vesicles coming into the Golgi apparatus are called transport vesicles. The vesicles leaving are usually called secretory vesicles. So these vesicles carry things to the plasma membrane to take out of the cell. So the Golgi apparatus is sometimes compared to, most stores don't have them anymore, but back in the old days, or sometimes at Christmas, places have them, the gift wrapping department. You know, the protein is the gift, and then we're going to wrap it up. We're going to do something to it. So these layers of the Golgi apparatus are going to chemically modify our protein. A lot of proteins, after they get made at the ribosome, they are non-functional. They can't really work. They have to have some modifications made first. And one of the main places where those modifications occur is in the Golgi apparatus. So if we look at a complete picture of how a eukaryotic cell does protein synthesis, we start out with the chromosomes. That's where our instructions are. A copy of the instructions gets sent to the ribosomes. From the ribosomes, most proteins move inside the endoplasmic reticulum to move to their final destination. Sometimes they don't go in the ER if they're very close to where they're going to end up. Then a transport vesicle carries the protein from the endoplasmic reticulum to the Golgi apparatus. Then a transport vesicle or a secretory vesicle carries the protein to its new location. So notice if we look at these, the nucleus is a membrane-bound organelle. The endoplasmic reticulum is a membrane-bound organelle. The transport vesicles are membrane-bound organelles. The Golgi apparatus is a membrane-bound organelle. So lots of membrane-bound organelles, and we're not completely done with them yet. Now let's look at two of the vesicles that we see. One of the types of vesicles that leaves the Golgi apparatus and stays in the cell is something called a lysosome. Now remember we talked about hydrolysis reactions before. So there's that LYS again, and that means to tear something down, break it apart, unbind it. Now notice some, like ribosomes, chromosomes, lysosomes, and we'll see some more examples later. This means a little part. So there's a lot of things in the cell that end in some. So lysosomes contain digestive enzymes, and they contain digestive enzymes that carry out hydrolysis reactions. So there's that LYS again. Now remember, hydrolysis reactions are what cells do when they take large molecules and break them down into smaller molecules. They add an OH to one part of the molecule, they add an H to the other part, and then they split it in half. So what happens here? Well, big molecules like proteins are broken down to amino acids. Nucleic acids are broken down to nucleotides. Polysaccharides are broken down to monosaccharides. Triglycerides, remember those are the things we call fats and oils. Those are broken down to monoglycerides and fatty acids. So lysosomes are important in your immune system cells. There are cells in your immune system that basically eat bacteria and viruses. And then once they eat the bacteria or virus, they destroy it with all these lysosomes. So they break it down into small pieces. Because if you have a virus or a bacteria in you, and you take the proteins in that virus or bacteria and you break them down to amino acids, they don't work right anymore. So the bacteria or the virus can't really infect you. Now there are some cells where the cells, the lysosomes in the cells basically explode. And when they explode, all these digestive enzymes are released into the cell and they destroy the whole cell. So cells in your body do this sometimes when they're infected by a virus. If they're so badly infected, they can't be salvaged. They kind of commit suicide. The cell breaks its lysosomes open and it kills itself. Hopefully, it kills the virus along with itself. That's the idea. Another type of vesicle we find is something called peroxisomes. And we get peroxisomes from some of the vesicles that break off the endoplasmic reticulum. Sometimes they come from another peroxisome. Peroxisomes can reproduce, they can split in half. And if you're not using our book, if you're looking at something else, peroxisomes might be referred to as microbodies. That's the older name for them. And peroxisomes do two main things. They carry out a process called beta oxidation. And beta oxidation breaks down polyunsaturated fatty acids. Remember, unsaturated fatty acids have a double bond between two carbons. Polyunsaturated fatty acids have more than one double bond between carbon atoms. So usually they have two or three, but occasionally more. They also have some enzymes that give it their name. They have some enzymes that produce a chemical called hydrogen peroxide. Now, you might have hydrogen peroxide in your household. A lot of people use hydrogen peroxide as a disinfectant. If you get a cut, sometimes you'll pour a little hydrogen peroxide on it. Now, these organelles, these are found in your cells. They actually make hydrogen peroxide. And hydrogen peroxide is a cellular poison. So why are they making a poison? Well, because peroxisomes convert hydrogen peroxide into water. But the peroxide can poison bacteria cells because bacteria cells usually can't break peroxide down into water. So as a result, they get poisoned by it. They can't survive. So when you see that kind of bubbling that happens when you put hydrogen peroxide on a cut, that bubbling usually is the peroxide being broken down, because some of it's broken down into water, but actually some of it is actually broken down into oxygen, which is a gas. And so that's the bubbles that you see coming off of that. So when you see bubbles, that tells you that the peroxisomes in that area are working. So we have to think about all these different organelles, these 6, when we think about how a eukaryotic cell makes a protein. Now notice the peroxisomes that we were just talking about come from a transport vesicle that pinches off the endoplasmic reticulum. The lysosomes come from a transport vesicle that pinches off of the Golgi apparatus. And there are other types of vesicles that get made by the endoplasmic reticulum or by the Golgi apparatus, but these are the only 2 that we're going to worry about right now. So are there any questions about how a peroxisome or a lysosome works, or how protein synthesis works in a eukaryotic cell? Now keep in mind, when we talked about prokaryotic cells, we also started out by talking about protein synthesis. So this gives you a good hint that there are going to be a lot of questions about protein synthesis on the next test, when we have a test about cells, about another week or so. If there are any questions, we'll see if we can answer them before we leave, but if not, I will let you go for the day. Okay.